Bismillahirrahmanirrahim. Inshallah, we'll continue with our study of Mendelian genetics, which we started previously. Now, Gregor Mendel, Austrian monk who figured all this out, he studied various different characteristics of the pea plant. Previously, we were talking about the green color and the yellow color peas. But in addition to the color of the peas, for example, he studied various different aspects of these plants, and because he noticed that there were distinct characteristics. For example, he noted that there were two types of these pea plants. There were tall plants, and then there were short ones. And then he noticed that there were two types of the shape of the seed. There were the round ones, and there were like wrinkly looking ones. Even you can probably appreciate sometimes that when you get green peas from the store, sometimes they're round and sometimes they're wrinkly, and then yellow and the green peas, of course, and then the seed coat color. In other words, if you cut the seed in half, the seed coat is the inside of it is either green or inside of it it's either white. Okay. And then there's a pea pod shape, which is inflated, and there's one that's constricted or flat, like that air has been suctioned out of it or something like that. And he also studied the pod color, and then the green one and the yellow ones. And also the position of flowers. He noticed that there was a difference. For example, there's pea plants that had flowers that were axial, which means on the side like this, and they were terminal, meaning at the end of each little branch like that. So that's the position of the flower. So these are the various aspects of the pea plant that he studied. And for example, he noticed also that he took a tall plant and a dwarf plant. And then when he would cross them, and that is, he would cross the flowers, right? Because all genetic materials contained in those pollens and eggs, which are contained in the flower. So he would cross these plants, and then he would notice again that in the F1 generation, or the first filial generation, all the plants were tall. And then if he would cross these tall plant, this plant with another tall plant, the F2 generation, just like with the green and yellow seed, demonstrated a resurgence of the dwarf plant, in a 3 to 4 ratio, 3 to 1 ratio. 3/4 of the plants would be tall and 1/4 would be dwarf again. So this suggesting again that the tallness gene is dominant and the dwarf gene is recessive. Now, I have drawn this before. This is called a Punnett square. It's named after a gentleman who basically, for convenience sake, invented this method of trying to figure out the genetic makeup of the parents or children like this. So a Punnett square is just a model of trying to work out some of these genetic mathematical ratios, etc. Typically, like this, you put the different father's genes on the top over there, and the mother's genes are placed typically on the side like this. So you just see over here, and these would be the different alleles that would be that the father's genetic makeup is able to produce, and these would be the different alleles that the mother's genetic makeup would be able to produce. And this is Reginald Condell Punnett, and this is just names to honor him. And here would be an example of, now for in plants, what does it mean to fathers and mothers? Plants don't have like fathers and mothers type thing, right? So pollen is a male genetic material of a plant, so that's what meaning you would put the pollen genes over here, and the pistil, the female component of the gene, would be listed over here. So again, in this way, he worked out various different genes for these 7 traits that he studied, and he noticed that these were all dominant traits, being tall and round and yellow with the green coat and inflated pod shape and pod color, being green, and axial flowers. These were the dominant traits, and their complementary traits were recessive. So he studied all of these 7 traits, and he noted all the same genetic principles that were derived from these. Now, so it all started with these observations that if you take a yellow and a green pea pod, and in the first field generation all will be yellow, and then in the second field generation 1/4 will be green, as you have noticed before. Now, these parent groups, these are pure breeding yellow line. In other words, they're homozygous for that particular trait, and that's what a purebred is. And how do you get purebreds? You breed those particular plants or whatever until you always get these, and that's how you know they're purebred, so to speak. Isn't this simplistically speaking? Okay. So this is the experiment that we started out with, and you can see one of the reason these plants made very good subjects, so to speak, was because they produce a lot of offspring, so to speak, and the ratios are very easy to calculate in this way. Now, when you study like this over here, when you're studying yellow peas versus green peas, okay, you're studying one characteristics. Now, you can also study 2 different characteristics. For example, look at this particular experiment, okay? Here you have a round seed that is yellow, and then you have a green seed that is wrinkled. Now, these are 2 different traits. The first trait is whether a seed is round or wrinkled, and the second trait that's being studied here is whether it's yellow or green. When you're studying 2 different traits, that's called a dihybrid cross. Okay, hybrid is a combination of 2 different types of plant. Di means 2, so a combination of 2 different traits, right? So, and we're going to finish off by appreciating these dihybrid crosses. Okay, so now look at this. So if you do this particular dihybrid cross, okay, now look at this. The roundness of the seed is the dominant gene, and the wrinkleness is a recessive gene. In other words, if a seed is if it's wrinkled, that would imply that it's homozygous recessive for that wrinkleness. So being round is a dominant gene, which means that it gives its name, right? So the alphabet is R, and the little r will be wrinkled. And yellow, the gene or yellow color, is dominant, and that's where it gives its name to the alphabet. And the green is the recessive gene. So among the roundness versus wrinkleness, roundness is dominant, which is, I presume, why you see a lot more round seeds than you see wrinkled seeds. And wrinkleness, roundness is dominant, and wrinkleness is recessive, and yellowness is dominant, and greenness is recessive. Okay. Now let's just imagine if you take 2 parents, two plants with the following genotype. They are heterozygous for both of them. Okay. And it was 1 big R, 1 little r, 1 big Y, 1 little y, and both parents are like that. Now, how do you know that this is the case? Well, just hang on for a second. So let's imagine that they are both heterozygous for each trait like this. Okay, so this is the genetic makeup. Now, since there's 1 big R, so this would be a round, and 1 big Y, which is dominant. This would be a yellow. So this is a round yellow, as you see over here. Correct. Same thing over here. Big R implies it's round, it's dominant gene, and big Y implies it's yellow, and that's it, dominant gene. So both of these will be yellow and round seeds. Now, what happens if you cross these 2 plants? Now, what you notice is quite an interesting phenomenon when they are heterozygous like this. You'll notice that the ratio of the 4 types of seeds, you'll notice that there will be the next generation of plants will have 4 types of seeds, even though all these plants were all yellow and round. So they'll be round and yellow. They'll be wrinkled and yellow. They'll be round and green, and they will be wrinkled and green. So there will be 4 types of seeds noted, and the ratio will be 9, 3, 3, 1. They will be in that ratio. Now, why does this happen? Okay, why does this happen? That's what we're going to try to appreciate over here. Okay, now understand we are crossing 2 separate traits. Now, the greenness and the wrinkleness is hidden over here in these traits, right? Because they're recessive genes. Okay, you would never guess by looking at these plants that these plants could produce green or wrinkled seeds, because they'll all be yellow and round. Okay, so let's study this. And the first question then, because how in the world do you ever find these type of seeds in the first place anyways, right? How do you know, how do you make those seeds, so to speak, that are heterozygous for each gene? Okay, so we'll answer that question in a little bit. Now look at this, okay? Why does this happen in this particular ratio over here? Okay, that's what we're going to try to understand. Now, this would be a parent, right? This is the genes of the parent. Now, this parent, okay, is going to be over here, and this parent is going to be over here. Okay, like that. So now if you look at this parent over here, they're going to make gamete cells, or they're going to make pollen, right? And they're going to have 4 options. They're going to be able to make big R with big Y, big R with little y. That's big R, big Y, big R with little y. And they're going to be able to make little r with big Y, and they're going to be able to make little r with little y. Little r, big Y, little r, little y. So those are the 4 potential gametes that this plant will be able to produce. In other words, okay, they will be able to make this combination of gamete, this combination of gamete, this combination of gamete, and this combination. There are 4 combinations of these genes that they will produce. Now each gamete has to have 1 gene for roundness versus wrinkleness, and 1 gene for the color, whether it be yellow or green. In the same way, this plant will be able to make 4 types of gametes over here. Okay, there'll be big R, big Y, big R, little y. Okay, and little r, big Y, and little r, little y. Okay, as these 2 over here. So now these are the specific gametes that each of these parents will be able to produce. Okay, now if you cross these, now you're talking about hundreds of these plants, and you're going to cross them. Okay, if this pollen, okay, happen to meet with this egg, and when they fertilize, okay, when these 2 fertilize, they'll give you big R, big Y, big Y, right? And if these 2 gametes were to fertilize, they'll give you big R, big Y, little y. In other words, they will make something like this. The end result would be like that. I'm sorry. Look at this. So when these 2 meet, this is the result. Okay. When you write these, let's change the color here. When you write this, you always write the dominant gene first. Okay, if they're both the same, doesn't matter. When you write the dominant gene first, and then the recessive gene, and then you try to keep them the same. You try to keep the roundness, wrinkledness first, and then yellow and green second. So each trait separate. So dominant goes first, recessive second. Dominant goes first, recessive second, if that's the case. Okay, just as convention, so that people can follow. Now, if you do this, you notice this is all the potential combination of new plant genotypes. Okay. Then, for example, if this gamete were to combine with this gamete, that pollen goes with that egg, it will produce big R, big Y, little y. Okay, big R, that's this one, and little y. Okay, now this is all the possible combination. Now, how do we get this? First, okay, how do we make this table? You have to appreciate how we made that table. Okay. Over here are all the possible gametes from this parent, and over here all the possible gametes from this parent. Okay, and then we combine the pollen and the egg in this case to make all the possible new plant combinations. Now these are the genotypes. Now observe what phenotypes this will be. Okay, now this dominant gene, this will be a round. okay, and yellow. This would be round, and this would be yellow, round, yellow. This would be round, yellow. This would be round, yellow. All of these would be round, yellow. That would be a phenotype, right? Round and yellow. As you can see, there'll be a lot of round yellows. This would be round yellow. This would be round green. That would be this guy right there, round green. This would be round and yellow. This is another round and yellow. This would be round and green. Round, right? And green. That's this guy over here. This would be round and yellow. There's another round yellow. This would be round and yellow. This would be wrinkled and yellow. That's this guy over here. This would be wrinkled and yellow. That's that guy over there again. And this would be round and yellow. There's another round and yellow. And this would be round and green. That's this guy over here. And this would be round and yellow. Wrinkled and yellow. That's over here. All right, and this would be wrinkled and green. That one guy over here. So if you count them out, you'll see there will be 9 of the round and yellows. Okay, and then there will be 3 of these wrinkled and yellows, 3 of the round and green, and 1. This last guy over here is this one over here. Okay, so this is a dihybrid cross. We're studying 2 traits in 1 cross. It gets a little hairy only because there are different combination of gametes that can result from each parent, and you have to account for each gamete. Okay, so now how do you, as I said, how do you get this? This is what we just studied. We took heterozygotes for each gene, and then we got this. How do you get these heterozygous plants to begin with? Okay, well, what you do is you start out with purebreds like this. You start out with 2 plants that are purebreds. In other words, you cross until you always only get the desired trait. In this case, round and yellow, and wrinkled in green. Okay, so you cross. These are purebreds. Pure breeds. Like that. Okay. And then when you cross those pure breed, then you get this heterozygote plant over here, and then you take 2 of them right there, and then that's the F1 generation. Then you take 2 of F1s and you cross them. This would be the F2 generation. So this is how you actually get the original heterozygote for each gene. So these are called purebreds. And this is an example of dihybrid cross. And if you do that, and this is what we just did over here, this is the result. There will be 1 round and wrinkled. There will be 3 of these round and greens. Okay, there will be 3 of these wrinkled, and there will be 9 of those other ones. So it'd be 9, 3, 3, 1 ratio, as we have seen. Okay, so this is an example of a dihybrid cross, as we have seen. Okay. Now, the next thing we're going to do is we're going to talk about Mendelian genetics as it applies to some common human genes after a brief pause. Until then, wassalatu wassalamu ala rasulillah wa ala alihi wa sahbihi. Assalamu alaikum.